Assalamu alaikum. This is Ahmed Shams. Staphylococcal Chromosomal Cassette, or SCC. SCCs can be annoying to many microbiologists, but they are simply mobile pieces of DNA. So let's analyze the title, hoping to make it simpler. Staphylococcal means it can be found in any staphylococcal species, whether aureus or non-aureus, coagulase negative staphylococci. And chromosomal means it's not free and not in a plasmid, it's inserted in staphylococcal chromosome. And the word cassette means it's a case or a bag carrying many genes performing many functions. And for being a case, it has a space to carry extra genes or to lose them. And because it's mobile or transmissible between cells of the same species or different species, it must have different types and subtypes. To help you imagine and get ready for its detailed structure, I had to show you this diagram. It depicts how this mobile piece of DNA can insert itself in a staphylococcal chromosome, giving the cell new phenotypic trait, then may excise itself, turning the cell back into its original state. It always attaches itself in the staphylococcal chromosome at a gene called ORF-X, or Open Reading Frame X. This ORF-X is a conserved staphylococcal gene. It means it's present in all staphylococcal species. So it can be used in molecular techniques to confirm the presence of staphylococci in the sample. But remember, it's not part of the SCC. It's not part of the staphylococcal chromosomal cassette. It's part of the original staphylococcal chromosome. And to the right, you can read that the origin of this SCC is thought to be the coagulase negative staphylococci. It's believed that this SCC with its MEC gene originated in coagulase negative staphylococci, then was transferred to Staph aureus. And this theory has some evidence. The first one is that SCCs of coagulase negative staphylococci are more numerous and versatile than those of Staph aureus. They also discovered identical genes of Staph aureus SCC-mec in coagulase negative staphylococci. And the third strong evidence is that they observed SCC transmission from coagulase negative staph into Staph aureus in vivo. If you didn't like the last slide, you have a different one with different color but with the same content. Here we have a brief history of SCC-mec discovery. In 1959, methicillin was introduced in the clinical practice. 2 years later, Staph aureus responded by being methicillin-resistant Staph aureus. It was until 1980 when the molecular basis of methicillin resistance was discovered. A gene called MEC DNA was responsible for that. From 1980 to 1990, it was believed by many that MRSA is different species from MSSA, but this was refuted. In 1999, first SCC was sequenced. In 2001, SCCs were first classified as SCC1, SCC2. And in 2009, an international working group on SCCs was founded. They are responsible for monitoring and classifying different SCCs. And now we have 15 well-established, well-known staphylococcal chromosomal cassettes. This colorful piece of art is an SCC. It spans from the green piece on the left to the green one on the right. These green pieces are insertion sequences or direct repeats used by SCCs to recognize their insertion sites in staphylococcal chromosome. That purple area on the far left is an ORF-X, Open Reading Frame X. It's a gene of staphylococcal chromosome and not a part of SCC itself. But it's a gene of staphylococcal chromosome where SCCs love to insert themselves next to. The red, orange, and yellow pieces are 3 genes forming MEC complex. If they are MEC A, MEC R1, and MEC I, they will form a MEC A complex. If they are different, the MEC complex might be of other classes like class B or C or D or E. The light and dark blue genes form CCR complex, chromosomal cassette recombinase complex, which code for SCC recombination. Recombination means insertion and excision. This complex is composed of one CCRA subtype plus one CCRB subtype, or sometimes it's formed of a single CCRC subtype. So there are two genes, A or B subtypes, or sometimes one gene of the C subtype. In this example, we have the CCRA subtype 1 and CCRB subtype 1. These void J areas used to be called junkyard because they contain non-essential genes. But now they are called a more decent name. They are called joining areas, J for joining. They may carry antibiotic-resistant genes, and according to their content, SCCs are classified into subgroups. So MEC A or MEC complex in general and CCR complex classify SCCs into main types, while J areas classify SCCs into subtypes. Now we have 15 SCCs classified by the International Working Group on SCCs. They are classified from 1 to 15 in Roman numerals, and they are composed of mixtures of 5 MEC complexes, A, B, C1, C2, or E, and 9 CCR complexes arranged from 1 to 9. And as we mentioned before, the CCR complex could have two genes of A subtype plus B subtype, or could have one C subtype gene. Now let's have an exercise to check how to interpret an SCC type. In this example, we have SCC type 4 in Roman numerals. Between brackets, it's written 2B. 2 refer to the CCR complex type 2, which is composed of A2 and B2 genes. And the letter B refer to the MEC complex, MEC B complex. And according to the J areas, this SCC is subclassified into type 4A, small a. Please go up at the MEC complex and notice how MEC A complex was changed to MEC B, class B complex, when the MEC R1 gene is truncated or partially deleted, and an insertion sequence in green replaced the deleted part, and no MEC I gene is present. In this case, the MEC A complex is classified into MEC B complex, and this is how MEC complexes are classified. Finally, don't panic if you find an SCC diagram drawn in a mirror image. The principle is that ORF-X is downstream at the 3' end, while MEC R1 and MEC I genes, which are the regulatory genes of the MEC A, are upstream to it. Because as we know, the regulatory genes are usually on upstream to the main gene. That's why the red arrows refer to the direction of transcription from up to downstream. So both of these diagrams are correct. And please leave a comment of a topic you want us to discuss together.